Everybody. We just have a couple of people dropping in, so give it just a minute and then we'll go ahead and get started. So welcome everybody. This is the second webinar in our Dairy Foods Innovation at Cornell webinar series. This series is designed to introduce you to the dairy ecosystem here at Cornell that makes New York the best place for dairy foods innovation. Our focus today is on introducing the variety of dairy foods product and processing research happening here in the Food Science Department at Cornell. Our speakers will highlight processing technologies, innovative value-added dairy ingredients, utilization of dairy byproducts, and more. This webinar is being recorded and will be available on our Dairy Foods Extension website after the event. Please keep yourselves muted, and we will have an opportunity after each presentation for questions. So if you want to drop your questions into the chat box, feel free to do that anytime, but please do keep yourself muted during the presentations. Our first speaker is Dr. Samuel Alkane. Dr. Alkane is an assistant professor of dairy fermentations in the Department of Food Science at Cornell University. He holds a BS from the University of Maryland, College Park in cell molecular biology and genetics, an MS from Cornell University in food science, and a PhD from the University of Massachusetts Amherst in food science. Before returning to academia, Dr. Alkane worked as an R&D brewer for Miller Coors, and then went on to join Unilever as their lead food safety microbiologist for the North American ice cream business. He now leads the Alkane Research Group, which focuses on fermenting sustainability in the dairy industry by using yeast and bacteria to transform dairy byproducts into new innovative products or to control spoilage and pathogens in traditional dairy foods. Sam, I'm going to go ahead and turn it over to you.
Thanks for the introduction, Nicole. Yeah, and today, let me get my screen up here and running. Today, I am going to be talking and hopefully changing the way that you look at whey with some of the research that we've been doing here. And it'll be a little high level, but feel free to interrupt me with questions, things like that. And we'll keep it a little bit dynamic. And so when we talk about, right, our dairy, processing our dairy products, a lot of time, right, we're taking milk, in an area that I love, right, and we're transforming it into yogurt, right? And particularly, right, Greek yogurt has been very popular. It's a strained yogurt. And for every cup of Greek yogurt that we make, we get 3 cups of whey, right? And right, if you're thinking about the Greek yogurt industry, there are some older numbers there, but the Greek yogurt industry, there's around almost 800,000 metric tons of Greek yogurt were produced in 2015, right? And if you think that for every, right, one of those tons, you get 3 tons of acid whey. So you're getting nearly 2.1 million tons of acid whey being made in the US. It's a lot of excess whey. What are we going to do with that? It's not being utilized very well. And when we talk about our other dairy systems, right, we take milk. What about the other stuff that we do, right? We turn it into cheese, right? And for every pound of cheese, right, we're getting about 9 pounds of whey out of there, right? So we're making a lot of whey. And our dairy systems have really been, optimized and developed to extract value from protein. And so any way we take this skim milk and we dice it up, right, we got a lot of processes here that are focused, let me get my little laser, on extracting protein, right? Whether it's UF milk, right? Whether it's milk concentrates, whether it's our cheese, we take the whey from that, we pull out the whey proteins, and then we're left with what they show as these gray little sad circles, right? Which is our lactose byproducts, right? What are we doing with those? We are really kind of covered in a snow of lactose. And this is a challenge because, right, where we're going to use this, 70% of the world's population is lactose intolerant. So we can't easily put it into other foods, right? 30 to 50 million Americans are lactose intolerant. And so what we end up doing with all of this excess lactose is sending it, right, either applying it to the fields, feeding it to animals, sending it to anaerobic digesters or wastewater treatment. And there's very little value extracted from this lactose. And most of these systems can't even take up all the lactose that we have. So, what can we do with this? This has been a challenge for our lab. And we got to ask ourselves, well, what's in this whey that we're losing? So there's been some work out there. Carmen Moraro is going to be talking to you later. had done some work characterizing some of the whey that's out there, and they looked at this Greek yogurt acid whey here, as well as cottage cheese acid whey and some milk permeate from some companies. And what you see, right, the challenge with a lot of these whey streams is that there's no protein in it right now. There's a lot of those sugars that we talked about that people can't use, a lot of lactose. There are also some very interesting things that we're throwing away, right? There are valuable minerals, calcium, magnesium, potassium, right, that are important for human health, are just going down the drain. There are a lot of B vitamins that are present, particularly in things like the Greek yogurt acid whey, again, that are important for human health that are just being wasted. So the question is, how can we take, right, this very, base commodity byproduct, right, and then really transform it into something that people want? And so the work in my lab is all around then looking at how do we use microbiology to do that. And so some of the areas that we've been focusing on have been looking at alternative yeast that can take that lactose and turn it into something else. So where we've started is there's this yeast called Brettanomyces, and particularly Brettanomyces clausenii. Now, Brett is become a very popular yeast in the brewing industry. Traditionally, it was considered a spoilage organism. Sometimes it creates horsey or earthy notes in the beers or wines, but they realized that this yeast could also add a lot of complexity to beers. And it turns out one of the species of Brettanomyces can actually utilize lactose, but there hasn't really been much work with it, as opposed to some other yeast species like Pluveromyces, which can utilize lactose and have been used in the bioethanol industry. No one has really understood what Brett could do. And then the other area of looking at is, we talk about yeast in brewing. I'm an ex-brewer. Traditional brewer's yeast, or even, traditional vinter's yeast, Saccharomyces cerevisiae, it can't utilize lactose, but, right, if you add a lactase, you can hydrolyze that lactose and break it down into simple sugars for the yeast. And so we've done some work around kind of understanding how these fermentations behave. And so here is a study that just came out this year in Foods, where we took a look at a couple different yeast species, that Brettanomyces clausenii, that can utilize lactose. We looked at Pluveromyces marxianus, which can utilize lactose. And then we looked at three other species, this Brettanomyces bruxellensis, Saccharomyces cerevisiae, a strain that was isolated in Norway, and then a wine strain of Saccharomyces cerevisiae. These three can't utilize lactose, so we added in lactase. And we looked at their fermentation dynamics. So here, this is a graph of density over time. So as yeast ferment sugar and convert it into ethanol, right? Sugar is heavier than water. They convert it into ethanol. The ethanol is lighter than water, so the density of the liquid goes down. And so it's a very easy way to monitor these fermentations. And you can see that all of these yeasts, these, with straight lactose and these with lactase, were able to ferment out all the sugars that were present in Greek yogurt acid whey. This Brett bruxellensis did a little bit of fermentation and then it got stuck. And if we take a look at what was produced, right, that product, we ended up with beverages that were a little over 2% ABV. And if you look at the sugar content, there is no lactose left over. There was no glucose for the ones where we hydrolyzed the lactose. And you'll notice that they all use the galactose except for this Brett bruxellensis, right, which had that kind of stuck fermentation over here. But so they were able, right, to utilize the sugar and produce ethanol. And not only that, they were able to transform the acid whey into a more interesting beverage for consumers. I don't know how many of you have drank whey, but we asked a trained panel to kind of talk about the characteristics of the Greek yogurt acid whey, and you'll see, right, that it's very, very, right, got these aromas around dairy and lactic and sour. It's got strong lactic acid and dairy flavor. It's got a little bit of this kind of rancid cheese notes. It's a little cloudy, has a little bit of body, but it's mostly dairy. Now watch when we take a look at what those yeasts did to the flavor profile of this product, right? You can see that they completely transform it, okay? We suddenly, right, these yeasts they add these fruity, appley aromas. They add these fruity, appley flavors, okay? When you look at these dairy aromas, right, that were really high here for the acid whey, many of these yeast strains, right, completely pull those out, right? So you're not really noticing dairy anymore. right? Ditto with the flavor, right? This flavor, right, is all the way out here for the unfermented acid whey. And a lot of these, right, mute those dairy notes. And so now all of a sudden, you're making a much more interesting, much more complex beverage that then, right, is something that consumers might be interested in. And so we thought that that's really cool. And we've been doing a lot of work on further understanding those fermentations. Now, that's one area, right, where we've been looking, right, how do yeast actually transform this stuff? But what else can they do? So you notice, right, there were some different fermentation profiles between some of the species of yeast there. So we decided to take a deeper dive and understand how some of these yeast species utilize the sugars that are present in dairy, okay? And so what we did is we took that Brettanomyces clausenii, this Cloveromyces marxianus, and Saccharomyces cerevisiae, and we looked at how they fermented lactose, a solution mixed between glucose and galactose, so hydrolyzed lactose, glucose by itself, and galactose. And what you'll see here is that this Cloveromyces, right, it's happy fermenting all of these sugars, no matter what's present. The Saccharomyces cerevisiae, not surprisingly, is not happy when the lactose is present. It doesn't ferment it, so we don't see a drop in density, but it uses those other sugars pretty well. The Brettanomyces clausenii that we looked at, right, it can ferment out glucose very well. It ferments out the lactose slower than the others, but it ferments it out, right, and we get a product. It ferments out galactose, but when we have a mixture of glucose and galactose, interestingly, even though it can ferment lactose, when it's already split apart and it sees them, it gets stuck. Now this, right, is kind of interesting, right? What's going on here? What's going on with sugar metabolism and what opportunities does it provide is kind of something that we're interested in. And just here's a little bit more on the data where you can see here that the galactose is left over at the end of fermentation with some of our with our yeast, right? So that's a pretty interesting thing. So what can we do? So the galactose in it is an interesting opportunity because galactose is twice as sweet as lactose, right? So even though you have half as much, so lower caloric content, it's twice as sweet. So you still get some of the sweetness that you would have in dairy. It is a low glycemic sugar, so it doesn't cause an insulin spike like you would have with glucose, all right? And galactose is converted into hepatic glycogen in the liver, okay? And so if you are an athlete, you like to run marathons. Louise, you were talking about, one of our coordinators here was talking about running a marathon that she's training for. Right, when you hit a wall, that wall when you're running, right, you're just exhausted out, you've depleted those glycogen reserves. And so there's an opportunity here to use galactose as a way to replenish those reserves after exercise. And there are actually a few nutritional papers out there that have shown that as a recovery drink, galactose might actually be important for athletes. And I think this is something that we haven't taken advantage of in the dairy industry. And so using these fermentations, right, to either produce ethanol or to leave particular sugars, right, that have particular benefits for consumers are opportunities that we can use to make better beverages for the market. Now, on the other side of the fermentation coin, I'm an ex-brewer, so I'm always interested in ethanol. But some of you may also be familiar with other products that are out there like kombucha, which I call cowbucha, right? So in kombucha, what we're doing there is we've got this mass here. It's called a SCOBY, a symbiotic culture of bacteria and yeast that converts sugars into acetic acid. And it's almost in some ways similar to vinegar production. And what essentially is going on there is that Saccharomyces cerevisiae, right, the traditional yeast we use for brewing, takes sugars, right? So if you also think about cider making, right, or vinegar making, it'll take like the sugar, say in the cider or in this tea beverage that they use for kombucha, and it converts it into ethanol. And then another bacteria, Acetobacter, takes that ethanol and converts it into acetic acid. So it's a two-step process. And this first process happens when there's no oxygen present. And then when there is oxygen present, right, this Acetobacter then takes that ethanol and converts it into acetic acid. Well, what's the cool thing is, these are two organisms. Well, what two can do, Brett can do better, right? So that Brettanomyces clausenii, interestingly enough, one of the things that compounds that it sometimes adds to beer that adds complexity is acetic acid. And what we found is that actually Brettanomyces clausenii, and I don't know that there's any other organism that does that, can directly take lactose and under aerobic conditions when oxygen is present, it will then convert that lactose into acetic acid. And so we wanted to understand this a bit better in the lab. And so we took a bunch of strains from USDA, these Decara anomalas, which is just another name for Brettanomyces clausenii. We took our lab strain, Brettanomyces clausenii, which we'd actually gotten from a beer company called Omega Yeast. We looked at those, and then we looked at brucellensis, which is that species that couldn't use lactose, but is a very strong acetic acid producer. And then we looked at Cluvaromyces lactis, and we looked at, if we gave them lactose, what type of acetic acid production would we see over time? So you see day 0, there's no acetic acid production. And then you see, even by a week, right, we have some very high acetic acid production, almost 1%, right, by this Brett clausenii that we use in the lab and this USDA strain of Decara anomala. Some of these, it goes to show you that not all of these strains are strong acetic acid producers over time. Some are not. So it's important to know that. And then again, not surprisingly, right, that Cluvaromyces lactis really didn't produce any, and that brucellensis didn't produce any any acetic acid because it couldn't utilize the lactose. And if we look at the lactose content over time, right, again, we can see that, right, these strains are utilizing the lactose. It's not completely gone, but they're utilizing quite a bit of it in the production of that acetic acid. Whereas again, this Cluvaromyces or this brucellensis does not at all. Cluvaromyces uses it all, but doesn't produce acetic acid. And there are a couple strains of the anomala that apparently don't really utilize lactose and don't produce acetic acid. So again, there's some things to understand about this species, but there are a whole suite of them, right, that we can be looking at to be producing acetic acid from lactose and potentially creating some kombucha type beverages. And so right now, what we're doing in the lab is trying to optimize and understand these conditions. And so I'm not going to completely dive into these graphs, but the understanding here is we're trying to understand how things like the concentration of yeast that we're fermenting with, the amount of oxygen that's introduced through agitation, and then each one of these represents a different pH. So this is a pH around the pH that we'd see in Greek yogurt acid whey. This is a pH around what we would see for whey permeate, and a pH that we'd see around for milk permeate, and understanding then how all those factors combine and how we can then optimize for yield of acetic acid. And you can see, right, just visually looking, that there are differences, right, that are driven by pH. So we get, looks like here from our initial work, that a mid pH around with the whey permeate gets us our highest levels of acetic acid. We get pretty close with acid whey. The milk permeate is a bit less. And then, right, agitation, not surprisingly, right, introduction of oxygen is quite important to get that right to maximize the acetic acid production. Now, in that ethanol work, I also talked a little bit about, right, when we split the lactose into glucose and galactose, this Brettanomyces clausenii treats the sugars differently. So we've also been looking at, how do we optimize maybe a beverage, right, that has acetic acid and high galactose levels. So we've done some work, again, optimizing the conditions around, agitation, right, for aeration, time, inoculation rate, and pH to have high acetic acid production, but leave behind the galactose that we want. And then we can create, right, unique kombucha-esque beverages, right, that have a sugar that's better for you. The base, right, has minerals and vitamins that aren't naturally found in kombuchas, right? And then you have the acetic acid, which has some health benefits. So we make a better for you beverage out in the market for consumers to use. And I think these are opportunities for dairy businesses to take advantage of. And there's so many different ways, right, to slice these concepts. But I think this begins to show the groundwork, right, for a wide range of products that we can do, right? And we can lead the way to a new category of beverages. And now I'm going to just talk a little bit about how we've been trying to spin this out of the lab. So again, I'm an ex-brewer. I've been very interested in the alcohol space and a lot of the innovation that we've been seeing there. And so early on, we kind of took this concept of making a better for you alcohol and have translated it into this beverage that we're launching called Norway, okay? And kind of showing you the concept around this. It's around, again, a better for you, better for the planet, where, we are leveraging a wasted ingredient, which is this Greek yogurt whey, right? And that Greek yogurt whey is rich in minerals, right? So highlighting, right, these broad spectrum electrolytes. And right, this beverage, right, delivers an excellent source of calcium, good source of potassium, good source of zinc, good source of magnesium. right? There's no other, right, beverage in the alcohol space that does this kind of stuff. And then we kind of rooted in this tradition, this Nordic tradition. There's actually these some stories, some history out of Iceland, where they make skyr, which is their version of strained yogurt. And some of the old literature suggests that they would take the whey that was left over from skyr production and they would age it in barrels, and it would become alcoholic, and it would be a beverage that they drink. And so kind of rooting right around tradition, right? Taking tradition, kind of an artful story, and then the science to make something unique. And right, I think we're making some pretty interesting looking products. So we've got 3 flavors that will be coming out: a ginger, a mountain berry, and a citrus. And if everything aligns well, we've taken it out to retailers, and we'll be going to market at the start of 2022. And hopefully the idea here is that we can show dairy processors that there actually is a market here, and more people will get into this space and actually start utilizing their Greek yogurt whey and maybe their other whey byproducts to make these interesting beverages, because it is a space that's growing, okay? I don't know how aware people are of what's going on there, but the hard seltzer space has really exploded over the last 4 or 5 years. And the sour beer space in the craft beer market is also has also been growing. And just to kind of give you an example of this, all right, when we look at the craft beer market here, this line in purple, right, it's worth around $29.3 billion, and it's 26 billion barrels of beer, okay? The hard seltzer market in 2020 was around 6 million barrels, worth $2.5 billion, and it's expected by 2023 to reach almost the size of the craft beer market, which is huge, right? And like I'm showing, we could create beverages from dairy byproducts to compete in this space. And so there's a lot of money that can be made here, a lot of value that can be captured if dairy is willing to play in this space. And not just in the beer space, but also, right, I mentioned the kombucha space. There's a big space around better for you beverages, right, like tonics and things like that. And again, we're seeing a lot of growth and interest in that market. Pepsi bought Kavita back in 2016 for $200 million. Miller Coors, right, a large alcohol company, bought Clearly Kombucha for a large sum that was undisclosed. And that total kombucha market here in the US is expected to be worth $7 billion by 2028. And again, right, we have the science, we have the knowledge to build products that can get dairy in a space that we don't currently play in. And again, just one more, kind of financial example that I use to kind of, again, hit home the point of how we're leaving value on the table, right? We take milk, right, and we turn it into Greek yogurt, right? And right, a cup of that retails for around $1.20 on the shelf. If we were to take the whey, right, from that cup of Greek yogurt, it equals around 2 cans of Norway. Those 2 cans of Norway together retail for around $6, right? So there is more value at retail from the whey than from the yogurt that we're selling here. And right, we're just sending this down the drain to wastewater treatment, and we're feeding it to cows. So even, we may not capture all the yogurt whey into these type of products, but even capturing some of it, right, is going to return value for dairy companies. And I think this is important for maintaining, right, the economic sustainability of the dairy industry and the environmental sustainability, because we can't just keep throwing that all away. And right, it's not just Sam trying to, kind of go out to market. There are a lot of other companies that are starting up in this space and trying to push it. So for example, in Canada, we've got Vodka. What they're doing is they're taking milk permeate from a plant that's near them. They're fermenting it. The milk permeate comes at a higher level of lactose. They're fermenting that lactose into ethanol, and then they're distilling it to make this vodka. They've also actually launched a cream liqueur as well that is delicious. It's doing really well. I think they were the fastest selling hard liquor in the liquor stores in Canada. They're controlled by the government over there, but they're doing really, really well right now. In the US, we've got Wayward Spirits out of Oregon, where they're taking cheese whey permeate. They're fermenting the lactose in that, and then they're creating a neutral spirit that has been gaining steam. And then in the non-alcoholic space, again, we're seeing startups trying to play in this better for you kind of arena. And so we've got Superfrau here, which is launching a functional sparkling beverage where they're sourcing yogurt whey, blending it with flavors, carbonating it, and it's kind of based on a product. One of the founders of that company is from Austria, and they have some products out there that are a mix between juice and whey that are quite delicious, and they didn't understand why that wasn't in the US. And so this is going to be coming to market in early 2022. We've also got Spare Tonic, a pair of brothers. One's a chef, and they've taken yogurt whey. They have a proprietary fermentation that's non-alcoholic that transforms that whey into a tonic, and then they flavor it up. And this product is already for sale. If you're down in New York City or the Hudson Valley, you can find it at some of the natural food stores down there. And they are getting a lot of great publicity, and they're getting a lot of great traction. And so this is just an example of some of the ways that people are beginning to utilize whey. And I think that, you know, some of the larger companies out there really need to start focusing on using this product that is right at their fingertips that they're letting go out of the drink. So with that, you know, that's kind of my outlook on this. All we need to do, right, is follow the way. So if there are any questions, I'm happy to take them.
Thanks so much, Sam. We have about 3 minutes. So if anyone has any questions, you can either put them in the chat and we'll read them out loud, or you can also unmute yourself and ask your question out loud if you prefer that way.
Good.
So Sam, while folks are thinking or, you know, chatting, if someone wanted to sort of explore how this could work with their byproduct, you're sort of still developing these cultures and investigating how they interact with each other and with the product. What would you say as far as timeline if someone wanted to use those cultures that you're working with? How would they go about doing that?
Right. So with the cultures that we've worked with, the Brett or the lactase added yeast, I think a lot of that, right, we've gotten the dynamics there. So I think it now becomes more of a product development question as far as what are the flavor blends and things that you want to do. So you can either reach out to me, ask for a copy of the papers, and we can also, right, the point of being here at Cornell is we've got a pilot plant facility here where we're also, right, the idea is to help you guys develop these type of products. And so I'd say, give us a call. Let's do some work in the FPDL. And I think, right, within, 6 months, we could definitely come up with a product that I think would be ready, right, to then start testing with consumers and look into the consumer space and launching that. So.
Perfect. Thank you. Okay. If nobody has any additional questions, I don't see anything in the chat. Feel free to reach out to our presenters today directly by emailing them. You can get a hold of me or Louise if you need help with contact information. But everybody is, oh, Sam just put his email address in the chat. So feel free to reach out to anyone after the fact if you have any follow-up questions. All right, so we're gonna go ahead and move on then. Thanks again, Sam. Our next speaker is Dr. Ali Abbasbarad, Assistant Professor of Food Chemistry and Ingredient Technology here in the Department of Food Science at Cornell. Ali, I'll let you go ahead and take it from there.
Thank you, Nicole. Good afternoon, everyone. It's a pleasure to be here with you today and share part of our research related to dairy. My name is Ali. By training, I'm an organic chemist, and I joined Cornell Department of Food Science in 2015 after doing 6 years postdoc at Harvard University School of Engineering Applied Sciences. Currently, we have a team of scientists with multiple different backgrounds, polymer chemists, chemical engineers, mechanical and electrical engineers, physicists, microbiologists, food scientists, and material scientists. Our research focuses on ingredient, mainly food ingredient and ingredient functionalization, encapsulation of a lot of these actives or bioactives that I'll tell you a little bit about today, and microfluidic bioassays. And the last one or last pillar of research is focused on conversion of byproducts or co-products to value-added products. The first part that I want to, these are 6 short stories. The first part that I want to tell you about is emulsion, encapsulation, and designing release mechanism, which I'll tell you about, and then I'll also share with you a couple of examples. So usually we encapsulate or protect actives from a light, from oxygen, pH, undesired reactions, or thermal processes, and we actually want to design, trigger, and control release mechanism to deliver these actives, whether in food system or inside human GI tract. So it's a basically wide range of a delivery mechanism that we can utilize to deliver flavors, very sensitive enzymes, proteins, colorant, vitamins, probiotics, you name it. It's a wide range that we can actually try to deliver. Now, to create these emulsions or capsules, what we do, we in fact use conventional emulsion fabrication techniques. By this definition, emulsions are a mixture or dispersion of two immiscible fluids, such as oil in water or water in oil. For instance, if you take water and apply shear or shake it simply, you disperse it inside oil phase, you can create single emulsions. If you take these single emulsions and dump it into a bucket of water and again apply shear, you can create water in oil in water, or double emulsion, where you have a core of water and a shell or protecting layer of oil protecting that core material. Now, these double emulsions are basically our templates for encapsulation and protection of actives. What we can do, we can use a wide range of techniques for production of these emulsions: high shear homogenizer, high pressure homogenizer, which we have in our department both at pilot scale as well as bench scale, but also in our technique, we use Microfluidics, which is a platform to study these emulsions. What you're looking at here is one of these devices in action where we can create core structures. These are water drops encased or engulfed inside oil layer. And these are microcapsules that we can actually utilize for delivery of wide range of actives. One example is pH-responsive polymers. For instance, the polymers that are used a lot in pharmaceutical industry, but also they recently received approval to be used in food industry. These pH-responsive polymers, they can remain undissolved inside water in neutral condition, but once they reach to gut, to a stomach condition, or once they reach to a small intestine, they start to degrade, dissolve, and release their cargo. I want to show you just a couple of examples how we use these polymers to encapsulate and deliver actives. On the left side, what we are looking at is a movie of two different microcapsules encapsulating different dyes just for visualization. The green ones, they have a thin membrane, 500 nanometer, and the red ones, they have 4 micron, basically thick membrane. And once we change the pH or simulate gut condition, as you can see, the microcapsules start to degrade and start to release the cargo. Now, we can actually use the same mechanism, same type of polymers, and create a sequential release. For instance, in the right movie, what you're looking at is a combination of black and red, black and green microcapsules. Black ones, they are sensitive to a stomach condition. They are sensitive to pH around 2. And once we decrease the pH, simulate gut condition, they release the cargo. And when we ramp up the pH to small intestine pH around 7, they start to degrade and release the cargo. So we have a sequential release simulating what food experience, or these microcapsules, the experience when they get to small intestine and delivering the cargo. This is important because there are many food ingredients that we need to deliver. One example is Lactoferrin that we isolate from milk, and I'll talk about it. But there is a wide range of ingredients and bioactives that require protection from thermal processes, from light sitting in the shelf, from a long shelf life, oxidative processes that happens inside the food product. And once they are protected, we want them to be delivered and released in a specific GI location inside GI tract. So one example is probiotics. In this case, these are microcapsules. The diameter is order of 100 micron. And what you're looking at is a swarm of bacteria inside these are live bacteria inside these microcapsules. So they can proliferate and grow inside microcapsules. They remain stable. We can freeze dry them. We can put them inside a wide range of matrices, protect them from undesired, basically oxidative stresses. And once they get to the pH that is desired, the microcapsule, the membrane start to open up and releasing the cargo. So I want to give you one specific example on encapsulation and release. And the release platform that I want to share with you is essentially enzymatic trigger release microcapsules. So you all are familiar with Anato. People use Anato to color cheddar cheese and create orange cheddar cheese. Anato has two fractions. One is oil soluble and the other one is water soluble. So when you use Anato and create cheddar cheese, orange cheddar cheese, what you're left with is basically the whey. We learned from Dr. Alkane about the whey and whether it's sweet whey or basically acid whey. There are a lot of nutritious ingredients exist inside whey, and especially when we get these whey out or separated from cheese, people try to spray dry it to create powders, and this nutritious powder can be added to a wide range of products. So if you want to sell a basically whey, if it's colored, it's a very difficult task to do because people don't want to add, for instance, colored whey into infant formula or any other products that they don't have or they don't want conferring color into those. So what people do, they bleach the whey using, for instance, something like benzoyl peroxide, which can degrade the whey, the colorant in the whey, and create white whey. However, the problem is that it has a lot of challenges, health concern of flavor, because still we have proteins and basically fat or oil inside the whey. They can be oxidized and create off flavors. It's time consuming. And obviously there are health concerns for people who consume this. So what we have done in our lab using encapsulation platform that I just described earlier, we encapsulated Anato, a big fraction of Anato inside these microcapsules. The microcapsules are protected by a lipid layer, and the lipid layer is basically coated or decorated with casing. So what happens is that when you apply this or when you add this microcapsules that they're orange into cheese vat, what happens is that they stick to the casing because the coating is casing, and during the process they stick to the casing, and essentially upon maturation there are a lot of bacteria, there are a lot of probiotic inside which they secrete proteases and lipases. Proteases start to cleave off the protein coating and lipases start to cleave off the lipase coating. So the colorant is liberated in the cheese matrix and start to develop within the cheese block, which essentially delivers the colorant. So with this particular approach, we can mitigate partitioning of colored or Anato into the whey and only selectively color the cheese matrix. So here, what you're looking at is just SEM images or images of these particles where we can actually visualize. We can produce this in a larger scale. This is produced by our pilot plant spray dryer in our department. And this is a study where we actually mix these particles with milk and then we acidify it to apply basically to approach the isolated point of the protein, create coagulants, and then do centrifugation. Through centrifugation, we separate the whey and the curd, and what you can see here is the cheese curd becomes completely orange. However, the whey after freeze drying is completely white, even whiter than the time that you bleach it. So it means that you can create essentially white whey by selectively coloring the cheese curd without having the colorant bleeding or contaminating the whey itself. So this was one of the examples that I wanted to share with you, which we call it enzyme triggered microcapsule. Another platform that we have in our lab, as I pointed out earlier, we look into a wide range of byproducts in dairy industry and see how we can convert or extract value-added products from those. One of the main values, or one of the values that we have looked at, and there are companies that producing, in fact, lactoferrin, isolating lactoferrin from whey, is essentially is a very hot topic. Lactoferrin is a iron-binding glycoprotein. It exists in milk order of 100 milligram per liter. And what it does, essentially, it protects lipids and proteins and nucleic acids from free radical damage and oxidation, but also it regulates iron absorption. It's one of the rare proteins that very strongly binds to iron and protects it and transport iron to cells. So it's very important in terms of nutritional aspect, and it has antibacterial, antiviral, anti-inflammation activity. So essentially, the idea here is that how can we extract or sequester maximum amount of lactoferrin from the whey and essentially create lactoferrin powder and sell the powder so it can be used in supplements and a lot of different drinks. Again, a lot of companies, at least 4 companies to the best of my knowledge, doing this process, but the maximum capacity, the maximum sequestration that they get to is around 50%. So they can extract 50% of lactoferrin from the whey, from sweet whey. The rest, they can't recover it. What we have done in our lab, as I mentioned, I'm trained as a chemist, and we have a lot of chemical and polymer chemists, chemical engineers and polymer chemists in my team. So we designed polymeric resins. These polymeric resins, in fact, they not only match the performance of the resins that they exist in the commercial market, but also they can actually surpass. And right now we are getting up to closer to 80% of lactoferrin out of whey, out of sweet whey. And what we do, in fact, we use a wide range of monomers and the cross-linker. This is the cross-linker. So different ratio and design of the chemistry enables us to be able to target lactoferrin selectively. So what we do, we synthesize large range of resins and then select resins with maximum absorption toward lactoferrin. So we use pure lactoferrin in the first place to select those resins. Once we have done that, we take these resins and choose them by their maximum selectivity toward lactoferrin in a model whey mixture, because we are dealing with multiple different actives. Proteins that exist in the whey is not only lactoferrin, obviously. So we want to get maximum selectivity to extract only lactoferrin from that mixture. Once we have done that, the third step is desorption. We need to desorb the lactoferrin that is stick to the surface of resin and essentially recover lactoferrin from the resin. And the final resin candidates are essentially selected to use for isolation of lactoferrin from sweet whey. And optimization of the process is very important to get maximum amount of lactoferrin recovered from sweet whey. So this is just the SEM image of these resins. These are order of 100 microns to few millimeters, so we can control the size, matching the size or morphology of what is produced in chemical industry for a wide range of application. However, what we built in our lab and synthesized in our lab is essentially very efficient in selection of the lactoferrin. And what we do, we use HPLC and LCMS for quantification of the lactoferrin isolated after passing through these batch type of column reactor. So we can get up to absorption of lactoferrin up to closer to 80%, which is a huge progress already. And we are in the process of optimizing the type of resins as well as the condition, the pH range where we can actually recover maximum lactoferrin in our process. One thing I want to point out here is that lactoferrin is thermally unstable. We have apo and hollow lactoferrin, depends on the iron content that they have. So we not only extract lactoferrin, we have a different project that we work on, how to complex lactoferrin with other proteins to make them thermally stable so they can be added to beverages, and also they can be thermally processed and yet remain stable to show their antiviral, antibacterial properties. So in our lab, after sequestration and protection and stabilizing these proteins, we use our kits to look at their antiviral and also antibacterial properties and compare with lactoferrin that's separated with other means and to look at their basically biological activity. The next part that I want to tell you about is, again, looking at VEG and looking at lactose and essentially thinking about how we can convert a byproduct that is surplus that we have to a value-added product. In this case, what we are looking at is permeates where we can actually enzymatically convert to something called galacto oligosaccharide. It's a prebiotic. It's simply is a fiber that is basically you can create by putting lactose units together through an enzymatic approach, which is clean approach. And we can actually create this GAS. And if you look at infant formula or many other supplements, GAS and fructooligosaccharides FOS, they are added as prebiotic. These are food for our bacteria, gut bacteria, probiotic added to these type of products. So, we've worked with multiple companies. I think, in this case, Carl Moody helped us to get some samples, and we started working on this project. And now we filed patents, and also we have a couple of publications out already. We designed, in fact, glass beads or very large spherical beads where we immobilize enzyme in the surface. And what we can do, we can feed these to these enzymes and they convert the lactose to galacto-oligosaccharide. I'm just showing you the schematic, how it looks like. Very simple. We have peristaltic pump. We have also glass beads with the enzyme filled inside the reactor. And essentially the feed is fed from the bottom, and from top, we can collect the product and then measure that or quantify that with HPLC. So we can create a good or high percentage of gas, which in this case, we get to 55% conversion by feeding lactose-rich products such as permeate to the reactor and get basically the fiber for addition into a lot of different supplement type of products. The last one that I want to share with you is conversion of lactose to emulsifiers. So not only we can create value-added product, edible value-added product from lactose, we can also create emulsifiers or detergents that they are readily biodegradable and non-toxic. A lot of emulsifiers or detergents that we use are essentially made from more than 95% are made from petroleum-based chemicals, and there are a lot of different research shown that they can also be toxic for skin, for hair, for body, and at high dosage. And what we are trying to do is to leverage lactose and convert it to something that has similar properties as other emulsifiers, but also is biodegradable, non-toxic, and also has antimicrobial properties. So again, here we took permeate and fatty acids, and through enzymatic approach that we develop in our lab, we can create a library of emulsifiers. You can imagine if you change the size of this fatty acid here from lauric to palmitic, or for instance butyric, you can create a wide range of different emulsifiers with different water or oil solubility and cleaning properties. So we designed this process of lactose with palmitic acid, and essentially we created this process to be very clean, green, as well as clean label. Well, what we do is essentially in 5 steps, we can create emulsifiers that are based on lactose and fatty acids, and we evaluate their functionalities such as foamability as well as emulsifying properties, and compare those with span, tween, as well as a wide range of surfactant, SDS, sodium dioxide sulfate, wide range of surfactant that exists in food as well as cosmetic industry. So I hope that I could deliver very briefly deliver these messages that we in our lab, in our team, in our department, what we do, we use encapsulation and emulsification technology to encapsulate and protect and controllably release actives into a wide range of application as well as GI tract. One example that I showed you was coloring of cheddar cheese. We could selectively color cheddar cheese, but also I want to share with you, I'm serving as a scientific advisory board for Bill Gates Foundation on micronutrients and fortification. And lactoferrin is, for instance, one example I told you that we can use for iron fortification, but there are a wide range of other actives that we can actually use through encapsulation and control release mechanism for fortifying food. We learned that we can actually isolate lactoferrin with a higher amount of efficiency from whey. And as I mentioned, we have a different project in looking into a stabilization of lactoferrin and its thermal property, working with one of my colleagues, Dr. Rizvi. We are looking at how we can encapsulate it further and render its basically thermal stability expanded beyond certain temperatures. Also looking at whey impermeate, we convert lactose to prebiotic, also emulsifiers. So these are some of the agencies and companies that helping us in supporting our research: NDC, National Dairy Council, Dairy Promotion Board of New York State, USDA, NIFA. And this is our team. In fact, the work that I shared with you is a hard work of my team members. I always get the pleasure of sharing the results with you. None of these are done by me. It's only just the hard work of talented people in my team. So I would be more than happy to take any question you might have. This is my email address. Feel free to reach out to me if you have any question later on. Thank you very much.
Thanks so much, Ali. That was really great and really exciting. I love the term library of emulsifiers. That's fantastic.
Thank you, Nicole. Thank you.
So as Ali said, please reach out directly to him if you have any questions. We do have just a couple of minutes before we move on. So if anyone had any burning questions, you can put it in the chat or unmute and ask now. And I will give you just a second to do that. Ali, I'm wondering, as far as the Lactoferrin, do you have an idea of what the market potential for that product is as far as value?
Absolutely. To the best of my knowledge, talking to both nonprofit agencies as well as talking to companies, it seems that there is a huge demand for it. The challenge is that the source is limited, basically, right? So you need to go through a couple of steps to separate it or isolate it from sweet whey, because you want the lactoferrin that is not denatured in a native state because of the biological activities and properties that it has. So I think that's one of the major pathways that we need to take to be able to sequester as much as we can from dairy industry. I'm aware that there are a couple of biotech companies that they are trying to produce these through microorganism, right? So through CRISPR-Cas9, they genetically modify and try to produce lactoferrin through microorganism. It seems that the market is huge. Right now, for companies, they are producing these and commercially available, but they are very expensive because of the basically lack of enough resources.
Yeah, okay. Wonderful. Thank you so much, Ali. And I think we're going to go ahead and move on. Again, just reach out directly to Ali if you have any questions or through Louise. Our next speaker is Dr. Carmen Marrero. Dr. Marrero is a professor of food processing and chair in the Department of Food Science at Cornell University. She received both her BS and PhD in food engineering from the University of Galactus in Romania. Her teaching and research program at Cornell focuses on using engineering principles and novel processing methods to improve food quality and safety, with a special focus on dairy foods. Carmen, I'll go ahead and hand it over to you.
Should unmute so that people can hear me. Good afternoon, everyone. Thank you, Nicole, for the introductions. Thank you also for the invitation to participate in this seminar, and greetings to all participants. I was happy to see some familiar names and certainly glad to see everyone here. In the next minutes, I would like to give you a brief overview, like my colleagues did, on the technologies that we're working on and how they can contribute to innovations in the dairy industry. I'm going to run you through a number of things that we are doing, but as the others also said, feel free to reach out if you need additional details. So in all the work that we do, we try to keep in line with the trends in the industry and the consumer trends that we see on the market. And while these change a little bit year to year, I think we can all agree that some of the key traits that consumers expect from products, including dairy products, have stood the test of time, and they still relate to freshness, safety, nutrition. High protein is here to stay for some time now. Clean label, innovation, sustainability, and we're trying to use technologies and innovate and come up with innovation solutions that that cater to these key traits. So one of the processes that we've used for a long time in my group is membrane filtration that we've used in a number of ways, but in particular as an alternative to heat treatment for removing microbial cells from milk. And more recently, we're also using this technology to remove water from milk and other fluid products for concentration purposes. And the reason is that we intend to make products that have this fresher taste, that maintain their taste and nutritional value, but still deliver the safety and the shelf life that are absolutely critical for all products, but dairy products in particular. So as one example here, I want to describe a little bit our work on microfiltration. This is something that we've done for a number of years. We are using microfiltration to remove bacterial cells and somatic cells from milk. In my group, we're doing this at low temperature. So really, this serves well the freshness of the product, because there will be very minimal heat treatment of the product. So you can obtain something that is similar to an ESL product, or it is an ESL product, but that has undergone only minimal pasteurization. So what we were able to obtain with this is some substantial reduction of microflora. And if we look at bacterial cells over time in milk that has been treated with membrane filtration and minimal pasteurization, you're looking at the green dots on this very busy graph here. You can see that we achieve basically months of microbial shelf life for the product, which is something that companies are starting to pay very close attention to. This technology is not that new in itself, but the application is really something that is currently being explored by a number of companies in the US. Other countries have been ahead of us a little bit. In Europe and Canada, there are more companies that are using this technology, and we're hoping to see more of it used in the US. As I mentioned, we also turned our attention to the use of membrane filtration to replace thermal concentration. Milk has a lot of water in it, and if you want to transport it long distances or to convert it into other products, you need to remove water. That has been traditionally done using thermal evaporation. It's largely still the technology of choice for dairy processing. However, we all know that there are changes associated with thermal concentration. So the industry has turned a little bit towards the use of membrane filtration, which really removes water physically by either applying pressure, which is the case in reverse osmosis. And some companies, some dairy companies, are now using RO to concentrate products. One challenge here, besides the fact that it takes quite a bit of pressure, so you need these very large pumps, one challenge is that you run into membrane fouling pretty soon, and instead of getting to those high levels of concentration, 40, 50 Brix you can reach sometimes in thermal concentration. With RO, you can only get into the 20 Brix range. And To address this, we started working with forward osmosis, which is a little bit of a different type of process that uses actually not high pressure, but the difference in osmotic pressure. So you have to have a concentrated solution of sugars or salts on the other side of the membrane that will actually pull that water from your product, and that's how the concentration occurs. And because you don't use these high pressures, you have less membrane fouling. And this is a very promising process. It's used currently mostly in water purification, some energy creation, and slowly it's making its way into the food industry. We have done some work with funding from the National Dairy Council, where we proved this can be used successfully for concentrated milk, both skim milk and whole milk. You can see here the concentration level that we reach in terms of bricks. A little bit better with skim versus whole milk. The fat always has its role and causes a limitation. So certainly this is something that can have promise. We also used it for other products, and one in particular is a product that Sam Alkane talked at the beginning of our seminar here, and that is acid whey. Acid whey has a lot of water in it, or actually very few solids, depending on how you look at it. And as you probably very well know, it's a very challenging product to handle. And if you want to concentrate and dry it, it's even more of a challenge because of the amount of lactose that it has in it, and the product gets very sticky, and the acidity also causes issues. So we have used membrane filtration to concentrate. This is a proof of concept. We did some work with funding from the New York State Department of Environmental Conservation. And what we did in this case was actually to combine the benefits of these two processes that I described earlier. Reverse osmosis does really well in the low concentration range. You can remove relatively quickly a large volume of water from your product and then finish it off with forward osmosis without having to deal with those limitations caused by membrane fouling. So we are able to reach concentration levels that are comparable to thermal concentration in a reasonable amount of time. Of course, with membrane filtration, you can scale it up. You can just add more surface area of membrane if you want to speed up the process. So this is something that we actually were able to do, and we're using this combination process for other products as well, not just acid whey. This, as I mentioned, was proof of concept. One idea here is that what if we go the extra step? We remove water to a point where you can actually make milk powder. How would you make milk powder using a different type of process? And we've turned our attention to a new method of drying, that is microwave vacuum drying. I'm just showing here the flow diagrams for traditional milk powder processing. And on the right-hand side, this alternative type of processing milk into powder, where you use membrane filtration as a concentration step and then finish it off with a drying process that is different than the spray drying. Spray drying does very well, but if you're a smaller processor, you may not want to invest in this large spray drying equipment, and then you can use something that is a little bit more versatile, more flexible, and we looked at microwave vacuum drying. We started working with this technology just a few short years ago, about 2 years ago, 2-3 now. I want to mention that the person who is the expert in microwave vacuum drying in my group is Josef Dumpler, research associate in my group, who is on the call today. So I'm going to defer all hard questions to him, but a lot of the work that I'm showing here is his work. So we're using this dryer that is located in our Geneva pilot plant for our microwave vacuum drying experiments. This is a batch pilot scale equipment. There is also equipment available that's 10 times the size of this one, 10 times the capacity, I mean, and that is more suitable for commercial applications. What the proper comparison probably is in terms of drying is not necessarily with spray drying, but rather with freeze drying. So it's a much shorter process than freeze drying, and it has some comparable product quality. Obviously, with freeze drying, you get better quality because you are not going to heat the product, but it takes a very long time and it's very expensive. So we are looking at this as an alternative way to dry various dairy products. We've worked with concentrated milk. We're doing some interesting work. Joseph is doing that now with cream. So in terms of our approach to this, we're using both experimentation, where we really take the systematic approach to optimizing the process, looking at the different processing parameters, different product parameters. This is a complex process that actually combines microwave energy and vacuum. So there are many intricacies of this process, and obviously the product characteristics also play a significant role. So this is something that takes quite a while to understand, but we can do it in a systematic experimental way, as well as using predictive models. And Joseph has developed this model where you're looking at product properties and also drying conditions to predict what type of time scale do you need for drying, what would be the drying rate. So that would reduce significantly the number of experiments, the level of experimentation that's needed. So you start already at the place where you know what to expect, and then you just conduct a limited number of experiments to optimize your particular process. And that has been working very well because he's spent quite a bit of time optimizing it. And now when he has to deal with a new product, that is relatively straightforward and easy to do. So we use some of this knowledge to actually dry other products. What are we drying now? Well, cheese. You may be familiar with the Moon cheese. That is a commercial product. We obtained some funding from the National Dairy Council to look at the process in a systematic way and create a range of products, because you can get something that is a puffed product like you see here, where you can get crisps from cheese. So this would be putting the dairy industry in that snacking aisle where we don't really have a presence as much. So this is what we've been doing as part of this process. It's an ongoing process. Interestingly enough, depending on what you start with, you can get a range of products. This is me keeping one of these big products in my hand, but clearly this is, while this looks interesting and fun, we jokingly call this horse snacks, not human snacks. We're focusing really on the human snacks that you see here on the left-hand side. So one of my grad students, Bailey, is now working on optimizing the process, again, to determine what are the energy and pressure levels that optimize expansion, because it's not a straightforward relationship. And there is this interconnection between the process and the product. And we're understanding, again, how drying can be sped up and what conditions do you need to obtain a product that has the lowest moisture content, the lowest water activity possible. So it is, in the end, a shelf-stable product, not just microbiologically, but also from a sensory and chemical perspective. So this is something that, as I said, we hope will help the dairy industry approach the shelf-stable snack market, which is a very strong, and still growing market at the moment. And not many people find, not many dairy products can be found on that shelf. So if you're interested in learning more, please let us know. Last but not least, I'm going to talk about some exciting work that we're doing using probably the newest technology that I'm going to talk about today, that is high pressure processing. In itself, it's not that new anymore, but clearly it is in its infancy compared to other technologies. And you may already consume products that are made using high pressure processing. Probably the best example that you can relate to is guacamole. That has been the first product, the first commercial product, but there are a variety of juices now and even meat products that utilize high pressure processing to kill microorganisms. So it's currently mostly used as a non-thermal alternative to pasteurization. So it kills vegetative cells. We are using it in a different way. Yes, when you use high pressure processing, you will kill vegetative cells, and that helps as a safety and shelf life assurance step. But we are using this technology to create novel structures, because high pressure can help unfold denatured proteins, and then when you remove pressure, they will aggregate. So you form these gels. If you have a high enough protein concentration, that can be used to create novel, interesting products. And this really can create a different path from milk to a finished, completely new type of dairy product that is not available on the market. We have looked at it again systematically. We're scientists, we do that, but really in the end, this is what we can get to. Well, this is a milk protein concentrate that has been high pressure treated. Now, don't imagine that it comes like that as a block construction out of the high pressure unit. This is something that one of my grad students did just to show the strength of the gel that you can get. If you just treat the protein concentrate with pressure, you will get a relatively soft gel with a limited shelf stability. However, you can improve that strength and the stability by decreasing the pH, by adding calcium, by adding granite. Does this sound familiar? Yes, that sounds like cheese making, correct? So we are able to do that. We're not proposing here an alternative method, to cheese making, because we know the complexities of cheese making, but I think we can penetrate this snack market in a different way. This time, this will be refrigerated, not shelf stable, but the beauty of it is that if you add flavorings, if you add vitamins, taste components, they will not change. Actually, because the high pressure process affects these big molecules, but it does not have a detrimental effect on the small molecules. So you can have something with high nutrition, and also in this case, as you can see, this is just milk protein, and you can add certain elements to it and make a high value protein snack with this. The takeaway message that I want to leave here today is that by combining new technologies, new ingredients, and new ideas, you can end up with new and interesting opportunities and innovation in dairy. And some of these actually can help improve the sustainability of dairy processing. They use less energy than thermal processing, and some utilize better resources than traditional processing. These are all exciting developments. Obviously, the test of a technology or of an idea is whether or not it made it to market. And this translation of research into industry requires a partnership. And I hope that some of you will be interested in some of the ideas or all of the ideas that myself and my colleagues are presenting to you today. And I encourage you to actually contact us if you want to hear more. I would like to acknowledge the funding that was that made this work possible. That ranges from the New York State Milk Promotion Advisory Board, National Dairy Council, New York State Department of Environmental Conservation, USDA, and obviously I want to acknowledge all the people who did the work. I'm just talking about it today. I cited them throughout, but just want to give a shout out to them again. And with that, thank you for your attention. And you also have my contact information here in case you have questions. Thank you.
Thanks so much, Carmen. If anyone has questions, again, you can throw them in the chat. you can unmute yourself and ask questions directly to Carmen. We have a couple of minutes still available. Carmen, I actually have a question about the model that you and Joseph have developed for the vacuum microwave drying application. Is that something that would be available to companies to use? Is there a user interface or a way that could be transferred to a company for predicting how that technology would work with specific products?
Excellent question. I think that the direct answer is that this will become available as soon as we publish this work, Joseph has put together the manuscript, and we are going to submit that for publication soon, and then it becomes available. But you can also reach out, and we'll be able to discuss that with you more. And as I said, Joseph's also on the call today.
Perfect. Thanks so much, Carmen. All right, if there's no other questions, then let me introduce our last speaker. Dr. Sai Rizvi is the International Professor of Food Process Engineering in the Food Science Department here at Cornell University, with adjunct appointments in the Cornell School of Chemical and Biomolecular Engineering and in the Department of Chemical Engineering, University of Toronto. Dr. Rizvi's research has advanced the food engineering field by pioneering novel extrusion, membrane separation, and supercritical fluid technologies of utility in food and dairy manufacturing. His teaching and research focuses on integrated applications of processing and engineering principles and practices for the delivery of better quality food and international development. Dr. Rizvi, I will turn it over to you.
Can you see my slide?
Yes, we can.
You can? Okay, wonderful. So I'm going to talk about engineering processes for dairy foods innovations, and I'd like to focus on four major categories, give you a little tip of the iceberg treatment to really get you interested in it. And then if you need more information, and indeed I'll be happy to get into more details. So let's get in the first part, extrusion processing, then followed by supercritical fluid-based processing, bioactive delivery systems, and cryogenic freezing. So first thing is dairy industry is under a challenge. It's a hard time for dairy industry because there are alternative sources of products that are biting into the domain of the dairy industry. And you are very familiar with how the milk sale is affected. You can buy ice cream, cheese, butter, all kinds of things from plant-based products. So this has put a little kind of a challenge to us to how to help the dairy industry do better. And of course, it offers many opportunities. Milk protein is the gold standard for protein, but it has been limited to gym culture. We haven't really capitalized on it as much as we should. And there are some movements in that direction, but I think that there's a scope for more. Lactose, a byproduct, limited utility, looking for new options. You heard about oligosaccharides, being converted, from lactose, and that's another area of potential activity. Fat, a source of diverse lipid profile, 400 different fatty acids. No other fat source can match that. And it has unique flavor and functionality that we need to be exploited. So let's look at some of the things, So average US per capita, price for protein is really listed here. If you look at milk, when you buy milk, you are paying roughly about 5 cents per gram of protein. If you look at nutrition bar, you're paying about 20 cents per gram of protein that you buy. So why not to convert milk protein into more NutriBar at an affordable and attractive price with added benefits. So opportunities are in everywhere. Look at the infant formula. There are a lot of products being introduced in this category globally, and even in the US. Every year we are seeing many new introductions in baby formula, and milk was designed by Mother Nature for babies. So what could be a better food for babies and toddlers than milk? But other categories are barging into this domain. And you can see here now the first baby puffs. This is a replacement of the conventional baby food coming in a jar that you shove into babies, and in 15 minutes they're done. Now babies learn to put everything in their mouth when they're 10, 15 months old, 8, 10 months old. And this companies have generated this Cheerio type products called Happy Puffs. These are extruded products. Baby can feed himself or herself, and it melts in the mouth in less than 30 seconds. So it's there's no risk of any choking or hazard from these products, and it has become very popular. Many companies are making these kinds of puffs. But if you look inside it, it's a very low nutrient density product, and it's all starch based. And if I look at the nutrition label, a serving size of 7 grams, carbohydrate is 6 grams, there's 0 protein. I mean, this is a product that, it's a matter of time when somebody, a medical doctor, will say, look, food industry is pumping hydrolyzed starch into our babies, and that's causing obesity. But it's the number one selling fried baby food in its category. So why can we do this with milk protein? Create in-mouth dissolving milk protein-based puffs. And this will not only help deliver better nutrition to babies, but also we can create a geriatric food or food for people who have dysphagia or swallowing difficulty. Can we create a product? And I was talking to a medical doctor at Weill Medical School, and he said this will be fantastic for people who have swallowing difficulty in the geriatric ward, because only thing is Ensure, which is refrigerated product. After you open it, there's a lot of liquid in it, and there's no solid food that could replace it right now. So anyway, this is an opportunity. So we looked at it, we said, well, why can we do this, make a milk protein-based puffs that dissolve in mouth in less than 30 seconds. So, but if you do look at cooking extrusion, which uses about 130 to 180 degrees, that is very good for starch-based products. You can dextrinize starch, and that will become more soluble in water. That's the key behind these Happy Baby Puffs that disintegrate in less than 30 seconds in the mouth. So for making the product, if you use this with protein, it's not going to survive. This is how milk protein concentrate 80 will come out, because the lactose and protein will go through Maillard browning, and this product is not going to dissolve in mouth in 30 seconds. So therefore, also, that's why you do not see any extruded products made out of milk protein exclusively. This milk is always added to these cereals, and when cereal consumption is going down, milk consumption has gone down with it as well. So we looked at it. I've been working on this technology for some time now. Extrusion combined with supercritical or high dense carbon dioxide, we call SCFX process. We've been at it for a number of years, and it decouples the role of water as a plasticizer and a blowing agent. And it's a green, low shear, low temperature platform technology that we have developed that can deliver this challenge of making milk protein based puffs. So using that, here is the idea behind it. So you look at, the same idea as a conventional extruder, except that we keep the temperature below 100 degrees C, and by circulating water, and we inject high pressure carbon dioxide as a blowing agent. And now we make carbon dioxide expanded products at low temperature and low shear. And another beautiful thing about this is that when you inject carbon dioxide, we can also control the pH inside, because carbon dioxide, when it dissolves in water, it forms carbonic acid. But the nice thing about this is carbonic acid acidity is temporarily induced in the barrel. As long as you have pressure inside the barrel and carbon dioxide is dissolved in it, you have carbonic acid. When it comes out of it, carbon dioxide gets out, you get your normal pH back. But this gives us a very nice bioreactor that we can do a little chemistry behind it, and to our advantage, and create products that normally will require change in pH, etc., that will have to be neutralized. But here we don't have to do that. So dairy products now can be extruded. So you can take now milk puffs, compare this with the steam puff. We have no browning, no Maillard reaction, nothing. So with this, we said, look at amino acids and lactose, this is what causes Maillard browning. But we know that low temperature and low pH induced by carbon dioxide will eliminate that. And that's exactly what happens. Second thing is, if you take milk protein and heat it under steam extrusion, you see aggregation, calcium ion bridging, hydrogen and hydrophobic interactions take place that what make it impossible to dissolve, and disulfide bonding occurs. This is happening under conventional steam extrusion. So we add sodium hexametaphosphate, very small quantity to chelate calcium and minimize protein interaction through imposed acidity. And we can show you that really steam extruded one versus an extruded one, you can see electrostatic interaction versus non-covalent and covalent interaction, all taking place at, with the steam extrusion and a small quantity of carbon dioxide. But there's an optimum quantity of carbon dioxide and acidity, which will minimize this. So this is what we did, and we eliminated sulfur-hydryl content, sulfur-hydryl bonding. As you can see, when we inject high quantity of, about right quantity of carbon dioxide, it's very similar to unextruded product. A little bit lower, but very similar. So with that information, and we did some simulation, but because if you add carbon dioxide and impose acidity, if you go below 4.6, it will, coagulate protein. But it so happens that if you look at this and do with milk protein in the system, then our pH never goes below about pH 5. So this is a very useful thing to say. If you have just water and carbon dioxide, the pH can go below to 3.5 or even lower. The reason it happens because protein has a high buffering property. It buffers the system, so the pH is not lowered as much. And therefore, we maintain really healthy pH of about 5, minimize chemical reactions, and take it to do the job that we want it to do. So based on that, we optimize conditions to microstructure. You can see the amount of pressure generated and amount of carbon dioxide we added. You can see we can really create a microstructure which will disintegrate, pick up water quickly and disintegrate in water. Having done that, then we create, here's a steam extruded one, different quantities of carbon dioxide. This is the high carbon dioxide content one. And you can see now, this is a commercial product on the left and our product on the right side. And you let it sit for about 30 seconds and it will dissolve, it will disintegrate. The structure will disintegrate, and it will be even faster in mouth because this is just water as opposed to saliva. The disintegration is even better. So after 30 seconds, you see a little bit more time. Both of them, this is a commercial puff starch base. This is our product, which is our milk protein. And you can see that if you take that and just, the force of the tongue will be enough to disintegrate it. Water has penetrated completely. So we have simulated that with the market sample and MPC sample, and you can see this is the commercial sample force required versus our MPC extruded in 2-bite test. It's very attractive. So here is a approximate composition of 7 gram serving. We can give about 4.5 gram of protein out of 7 gram, as opposed to commercial products. You can see 0 protein, and you have same amount of calories, but mostly from carbohydrate coming into the product, which has, which is commercial product is 6 gram of carbohydrate. We have about 1.24 gram of carbohydrate. So nice thing is it's a long shelf life, dry weight, low transport costs, no water addition needed for consumption, quick in mouth disintegration in less than 30 seconds, complete amino acid profile is MPC. It has the same ratio of casein and milk and whey protein as in milk. So only thing is it's lower in lactose. Consumer acceptable sensory properties, we just did that. Consumers cannot tell, much difference in terms of disintegration in 30 seconds. It happens very similar to commercial product. Elderly and dysphagia patients will benefit from it. And it's also, if necessary, we can, in the process, we can add other nutrients that may be necessary to make it even more complete, or targeted delivery to some affected group, or lactating mothers, or people who need more iron. We can add those things as well during the process. So there's a patent pending on it, and we have a commercial interest. We're talking to some companies who may want to take it up. Second part I would like to talk about is production of high purity phospholipid concentrate from buttermilk powder. You'll wonder why we want to do this. MFGM, milk fat globular membrane material, how to isolate phospholipid from that and see what it can do. So milk phospholipid, excellent source of excellent emulsifying properties. And the very important thing is, I mean, this is something we have to think about. Milk fat is also an emulsion in water, in milk. But this emulsion is so heat stable that you can boil sterilized milk, nothing happens to milk fat globule membrane. That has not been recognized, that this phospholipid is so unique an emulsifier, a combination of this. It has several phospholipids, sphingomyelin, phosphatidylcholine. It's a cocktail of several phospholipids. And Mother Nature has designed it and given it such a strong thermal stability that we have not exploited it. So inspired by that, we said, well, let's isolate it and from buttermilk powder and see what we can use it for. So high purity MFGM phospholipid concentrates are commercially not available. They're available, but not very at a reasonable price. They're expensive and not easily available. So we used our benign supercritical carbon dioxide extraction process. With pure carbon dioxide, we extracted some triglyceride, then we added a little bit ethanol and extracted phospholipids. So having done that, so if you take buttermilk powder, for example, the bottom line, and it has a 5.4% phospholipids. If I take that 100 gram and put through my process, we'll get 3 fractions. Fraction 1 will be the one that I extracted with pure CO2. Fraction 2, CO2 with ethanol. And fraction 3 will be what's left behind in the vessel. So that's the third fraction. So look at 2.5 gram, 4.3 gram, 93.2 gram. This 4.3 gram is where all the phospholipid concentrated. So we get now 76.2% concentrated phospholipids in 4.3 gram out of the 5.4 gram to begin with. 5.4 gram concentrated into 4.3 grams with 76% phospholipids. So this is a very highly concentrated phospholipid preparation. Now, highly concentrated using carbon dioxide technology that does not involve any organic solvent. And this is environmentally friendly. It's a green process, good potential for scale up, and it's a very heat stable emulsion it can make. Now let's use this to do something more useful. So probably you heard from Ali about generation of a stimuli responsive bioactive delivery system. So if you want to deliver something into the small intestine and colon, you have to protect it from digestion in the stomach. So some bioactive material or whatever needs to be delivered or probiotics into colon and small intestine, we have to protect it from the stomach. So there are different ways of doing it. One of the methods is liposome, an assembly of phospholipid-based vesicles. These are phospholipid bilayers, and you can embed lipophilic cargo in between here. And inside here is hydrophilic, water-soluble like probiotic and other things. So you can have both types of molecules, hydrophilic as well as lipophilic, loaded in this vesicle called liposome. And these are generally generated using organic solvents. So first thing we did, we have made it with no organic solvent involved, only using carbon dioxide. Having done that, then we coated it with either chitosan beta-lactoglobulin or eudragit. And we have this coated liposome that will be stable up to at pH, any pH below 4.6, nothing happens. So in the stomach, it survives. And then when it goes out into the colon or into small intestine, the pH is higher and it triggers the release and the load is delivered. But the bad thing about this is not temperature stable. This soy phospholipid based, this bioactive, these liposomes disintegrate when you heat it only to about 60 degrees. So for food applications, we need something that can be pasteurized or heated at least. So if that's one of the reasons why liposomes have not been very heavily used in food fortification, food applications, because they are not stable. So this is where our friend comes into play, this phospholipids bilayer. We should learn from Mother Nature and use the one that's found on milk fat globule membrane material, a cocktail of those phospholipids. And that's exactly what we did. And therefore, we extracted this and took the extract phospholipid concentrate we made into MFGM-based liposomes and coated it with uridage so it doesn't dissolve in the stomach and delivery into the colon or the small intestine. And if you look at this comparison of stability, here is the phospholipid. If you liposome made with soy phospholipid, you heat it 60 degrees, it precipitates. And you can see that in confocal laser microscope, these are the phospholipids and they aggregate and break down when you heat it to 60 degrees. Now, if you make it with MFGM liposome, they're very stable. You can see that you heat it to 90 degrees C and they're very stable. So this is something very unique and the dairy industry should really capitalize on it, the cocktail of phospholipids found in milk fat globule membrane material that have very unique properties not found elsewhere. So key finding is MFGM-based liposomes heated to 60, 70, 90, up to 90 degrees for 30 minutes were heat stable. And soy protein, they break down at just about 60 degrees. And excellent stability when tested in simulated gastrointestinal juices, etc. And they were, something that we are working with companies to see if we can commercialize it. Last part I'll just briefly mention is delivering ice cream on demand. So one of my favorite projects that I've been working on for 2-3 years. Ice cream, you can see that global ice cream consumption, per capita consumption in liters per year in Western countries is about 20 liters per year. You go to China and India and countries where heavily populated is less than 0.1 liter per year. And so if they start eating 20 liters of ice cream per person per year, milk will sell like gold. So the reason is because, cold chain, it's not easy to sell ice cream made at, at -8 degrees and then stored at -18 and then transported at -15 degrees C and then stored in a freezer. So because this cold chain is very expensive. So impact on environment, non-refrigerated truck spit out 30 grams carbon dioxide, this refrigerated 18-wheelers 96 grams. Energy intensive operation. Most of the time distribution and storage energy costs about 30 to 40% in ice cream making. And ice cream making, if you look at the history, not much has changed since 1926 when continuous freezers were introduced. There have been some incremental improvement on efficiency, etc., but the basic premises is still the same. So we looked at it, we said we can use Bernoulli's effect and Joule-Thomson effect and create a system where you take pressurized gas, pass through the system. When it comes from high pressure to low pressure, you can create -60 degrees stream of carbon dioxide coming from 25 degrees feed. I can feed 25 degrees carbon dioxide. It comes out here at -60 degrees, -70 degrees. When it comes through the nozzle, this is small constriction, velocity goes so high that it creates a vacuum here. Similar to the aspirator you probably have used in chemistry lab to speed up filtration in using Buchner funnel. Same idea. So this sucks in here. If you look at ice cream mix and flavor and color, we can pull it in by vacuum and mix it with -60 degrees stream of carbon dioxide and get instantly frozen ice cream. And this is what we did, generation 1, generation 2, and now we have the 3rd and 4th generation we have gone through. And here is one version that I'll play here for you, very quick shot. And you open the door, and here is your ice cream in 3 seconds, a scoop of ice cream in 3 seconds. Okay. Another version of the same thing using a little bit larger stainless steel system. Okay. And you can see in 3 seconds, you can give you a scoop ice cream. People like that. And you can see that when we look at the literature and the crystal size, which determines the quality of ice cream, The smaller the crystal, faster the freezing, the smaller the crystal, the smoother the texture. And you can see that this is our Cornell dairy ice cream, literature ice cream, and look at the size of the crystals in ice cream made so instantaneously, is less than 5 micron. So we have done this. We have a patent was given to us in 2020, and we have made a number of frozen products. Very interesting thing is we have made even frozen milk. Really, if you put a little bit sugar in milk and put through our system, it can freeze literally anything. We can make ice cream, we can freeze soda, we can do water, we can make carbonated ice. A lot of things can be done, but if you look at here, we really put a little bit sugar in milk and froze it. It was so delicious. Everybody loved it. It was so smooth, you know, that on your palate that you really wanted to eat more of this frozen milk. No need to do anything else to it. So with that, I thank you for your time and attention. I'll be happy to take any questions you may have, or will be available later on, or whatever is convenient for you. Thank you.
Thanks so much, Dr. Rizvi. Does anybody have any questions right now? Go ahead and throw them in the chat or unmute and ask. I love the ice cream technology. I want one of these in my house, actually. But from a perspective of commercializing this, where in that process are you? You said you just received your patent, and so what would the next steps be for commercialization?
Actually, you know, we just licensed it. The patent has been licensed to a company, a startup company.
Wonderful. It's exciting. Coming to a street corner near you.
Well, you know, like they say, there's a lot of things, you know, it looks good. Getting a patent is one part of it, but innovating it into a commercial product, it will take some effort, you know. There are some issues with supply chain and availability of carbon dioxide and tank and this and that, you know. So let's see where it goes. But I'm very hopeful it should be successful.
Very cool. All right. Well, thank you everybody for joining us this afternoon. Thank you to all of our speakers. Very exciting technologies and products that are being developed in the department here at Cornell. I'd also like to acknowledge the support of the Dairy Farmers of New York through the New York State Dairy Promotion Board, who have sponsored this webinar today. We do have one final webinar in our series that will cover the facilities and the services here at Cornell that are offered to dairy industry stakeholders in order to facilitate dairy foods innovation. So that last webinar will cover our food processing development laboratory, our sensory evaluation center, and our Center for Technology Licensing. And many of our speakers today have touched on those different services. So we'll sort of dig into those and see exactly what's offered to dairy industry stakeholders here at Cornell through those individuals. And like I said, that webinar will take place in December. We'll have an announcement coming out soon about the specifics on that webinar. But thank you all so much for your presence, and have a wonderful weekend.
Thank you.